Should we start? Good morning, everybody. Today we have Dr. Rocío Pérez Portela, who is an associate professor at the University of Barcelona. Besides being a brilliant scientist, she's also a very close friend of mine, so be gentle with her. Rocío started her career at the Universidad Complutense de Madrid. I don't know how to translate this. And then she moved to the University of Barcelona, actually, to do her PhD. And then, it was the toughest years of the crisis, I think. So all of us had to move around the world, and she actually moved quite a lot. She did postdocs at the University of Plymouth, also in Lisbon at the ISPA, and then back here with Juan de la Cierva in Blanes. And then again, back to the outer world, to Miami Rosenstiel School of Marine and Atmospheric Sciences. Did I say it right? Good. And so she started as a taxonomist of ascidians, but she quickly moved out of that very interesting topic to actually approach broader questions in ecology and evolution. So she has been studying phylogeography, connectivity, population demography, and other questions in very different organisms, all of them marine animals, or most of them marine animals. And now she started with a very dynamic group at the University of Barcelona studying adaptation in echinoderms, which is actually what she's going to talk about today. So please join me in welcoming Rocío. Thank you very much for coming.
So thank you very much for this presentation and for this invitation to talk today about echinoderms in a changing ocean, strategies for survival. So I will talk about that because during the last 7 years, I have been leading a research team in adaptation using echinoderms as a target group. So the objectives of our groups is to investigate the biological mechanisms that enable species and population to cope with environmental shifts, to predict the resilience potential to climate change. So we mostly focus on temperature and pH because, as everyone knows, ocean warming, which means increasing in average ocean temperature, but also an increasing in the frequencies of heat waves and ocean acidification are the two major threats for marine ecosystems, right? And they are actually both are global processes. They are related with the increasing of anthropogenic CO2 in the atmosphere, and a big amount of this CO2, the 30%, is absorbed by the oceans. So this CO2 interact with the seawater. I'm not going to explain all of that, but this mixing with seawaters at the end lowers pH, and it impairs calcifying animals to have calcium for building biostructures, right? So it has physiological effects on the species, on the marine species, but it also has a cascading effect on the whole ecosystem, right? And echinoderms, most of echinoderms are quite important species in marine habitat, especially sea urchins, because they are keystone species regulating algae populations and, in general, the whole dynamics within the marine systems, right? So we have been working on the concept of adaptability. This concept of adaptability includes adaptation of the populations, because adaptation happens at the population level. So how these populations along marine gradients, imagine that this blue-white gradient is actually an environmental gradient. So we have different populations of the same species but can live along these large marine gradients, right? So adaptation is happening at the population level. Due to selection, overgeneration, and at the end, it changes allele frequencies along the species and along the populations. I'm going to show you an example about the way that we work with the genomic data. So imagine that we have a sea urchin species along this gradient, and we have, for instance, one neutral gene. Because in general, we work with large number of markers, random markers, across the genome, and we need to identify neutral markers and markers under selection, right? So imagine that we are analyzing a neutral markers. We have here this population. Actually, there are no different populations, and every color is a different allele, right? So there are no differences in allele frequencies if gene flow is very high along the gradient. But imagine that we identify a gene under selection, and for instance, the yellow and the gray alleles have any kind of advantage at the white part of the gradient. So at the end, due to selection over generations, these alleles are increasing the frequency in some parts in some populations, right? So if you have a look here, what you actually see is a completely different structure here and here, right? But we are talking about the same individuals and the same populations, right? So they are the same. But here we are having a look to the neutral markers, whereas here, sorry for the people with the video, but here we are having a look. So actually, we are looking for the genes under selection. We detect substructure under that general genetic structure over populations, right? So this is important to understand before talking about adaptation. But within this process, within adaptability, we also have plasticity. And plasticity occurs during individuals' lifetime, right? So, and is the result of the interaction between genotype and the environment. And classically, it was thought that both processes were additive, right? So if you have high levels of adaptation with different populations and also large plasticity, you have higher adaptability. But it's not always true, because more recent studies demonstrated that populations very well adapted to a particular condition of a gradient, of an environmental condition, they actually have less plasticity. So there may not be summatory processes, right? And also it's important to highlight that adaptation and plasticity are not always easy to distinguish, right? Because in some cases, adaptive changes in regulatory genes allow adjusting physiological to match environmental conditions. So at the end, you have different patterns of, for instance, of transcriptomics, different patterns of gene expression, but they are not due to plasticity, but specific adaptations or specific mutations in regulatory genes. That's why in many cases, we cannot distinguish both processes. So what we do, what we actually do is analyzing different species of echinoderms along different gradients of temperature and pH. In temperature, we analyze latitudinal gradients over very large geographical areas, and pH you will see in CO2 vents in the Canary Islands, right? And we use a multidisciplinary approach using population genetics and genomics, also transcriptomics. I think everyone knows what is transcriptomics, but here we can see with population genomics, we can see differences in allele frequencies as explained before. And here with transcriptomics, we analyze the gene expression levels of all genes at the same time in a particular moment, so we can identify if there are differences in gene expression, right? And also respiration rates or oxygen consumption, which means it's an indirect measure of stress, right? Also reproduction patterns. Here we are talking about intraspecific processes. We also analyze some interspecific processes. Although today I will talk mostly about intraspecific processes. We also analyze the relationship between the host and the bacterial symbionts and the animals with the environment for feeding trophic plasticity. right? So now I'm going to show you some examples of our research for you to have a general idea about what we have been doing during the last years. So we work with abundant species, widely distributed along these natural gradients with high dispersal potential along the coast of Europe. And we combine different kinds of species. Here, for instance, you have Arbacialixula and Costinasteria stenospina. Both species are subtropical species. They live in the Canary Islands, the coast of Africa, and also the whole Mediterranean. All these species overlap in the Mediterranean, that's true, because we are working in Barcelona or very close to Barcelona. Paracentrotus lividus is a temperate colder species. We find it along the British Island and the Atlantic European coast, also the north of Africa, Canary Islands, and Mediterranean. And we also have other species called much colder species as Martasterias glacialis here, because Martasterias glacialis is very abundant in Norway and south in the Atlantic. It's also right to the Canary Islands and the Mediterranean. Obviously, our initial hypothesis, and as you can imagine, populations of Martasterias glacialis in Norway are very different than populations living in the Mediterranean because they are living under different environmental conditions, right? And this is actually true. So Marta Martínez-Huetes, in her PhD thesis, was working with a large number of populations of Martasterias on the European coast, and she identified a large number of genomic markers called SNPs, and she was able to identify over 3,000 genes under selection. But interestingly, all these markers were inside of chromosomic inversions. She identified 14 chromosomic inversions in the 22 chromosomes of Martasterias, which is a lot. And it's very interesting because adaptation is not only happening in some particular genes, but it's happening in very large parts of the chromosomes. You have here a picture for you to have an idea what is the meaning of an inversion, right? So just for you to see, I want to show you results from one of the chromosomes, only one of the chromosomes, chromosome 14. So here you have a Bayesian clustering analysis for neutral markers of this chromosome, right? So here every column represents a different individual. Every different color in blue here is a different cluster. And here you have the probability of each individual to belong to a different cluster, right? What we can see here in general for neutral markers is what we would expect in a species with the large potential dispersal of the larvae. So there are no significant differences among or between populations within major oceanographic areas, but there are differences between oceanographic areas, as we will expect it in this kind of species. However, if we have a look to the to markers under selection, in this case under selection of salinity, because we detected chromosomes under selection of salinity and temperature. So in this case, they were markers under selection of salinity. We have a completely different picture. So it's the same chromosome, it's the same species, they are the same populations, but we saw completely different structure. So the Western Mediterranean was different because levels of salinity are much higher in the Mediterranean Sea, right? So, but what we thought it was true for all colder species, cold species or temperate species is, well, strong patterns of adaptation is not always true, because we were working as well with Ophiocomina nigra. It's a brittle star, this one that you can see here. And we had samples from Sweden to the English Channel and also the Mediterranean Sea. We also had an interesting number of markers, and we didn't find any marker under selection of temperature or salinity. Actually, we had this structure along the European coast from Sweden to the Mediterranean. No genetic structure at all. So not all the species have that kind of strong patterns of adaptation or genetic structure, right? So no signs of local adaptation. So give me a second. But we wanted to go a little bit further about how these species, the different species, endure temperature increasing, what kind of processes are involved in the stress responses of these species to increasing temperature, heat waves, or climate change. So we decided to make experiments in the lab. So we take the samples at the end of wintertime in the Western Mediterranean, close to Barcelona. After a period of acclimatization, then we made experiments with controls with constant temperature, the temperature at the moment when we collected animals. And then in the treatments, so half of the animals were controlled, half of the animals were treatments. And we increased temperature from 16, 20, 23, 26, which was the average temperature in summertime in that part of the Mediterranean, right? And we analyzed both respiration rate or oxygen consumption as a measure of stress, and also in some species we made analysis of transcriptomics to analyze patterns of gene expression. And now I'm going to show you the results from different species. We have been analyzing 5 species. I'm going to show you the results of 3 of them. So these are the results for Arbacia, Paracentrotus, and Ophiocomina. So just to remind you, Arbacia lixula is a subtropical species, and from previous studies with population genomics, we knew that it has a very weak local adaptation patterns, right? Along the Canary Islands, South Atlantic, and Mediterranean. Paracentrotus lividus is a temperate colder species with moderate patterns of local adaptation. And Ophiocomina nigra is the last one that I show you, no patterns of local adaptation. And here in this graph, you have oxygen consumption or respiration rates. And here I show you controls versus treatments. In pink, here we have the controls. Light green controls, light blue here controls, and purple, dark green, and this blue, I don't know how to, blue, other blue, the treatments, right? And let's see the patterns, so the response, stress responses for these animals. In the subtropical species with weak local patterns of adaptation, we don't see any response at 20 degrees. Oxygen consumption is increasing at 23, significant, not very high, but significant, and very high levels of oxygen consumption and 26 degrees. So it's starting to stress. It's stressing a little bit. In Paracentrotus, a temperate species with moderate levels of local adaptation, we see stress from the first moment at 20 degrees. And the stress is increasing at 23, and finally, physiological collapse at 26. That means that at the end of the summertime, Paracentrotus in this area of the Mediterranean is suffering a lot, right? I didn't tell you before, all these experiments were made with animals collected in North Catalonia, in Cadaqués and Blanes. So it's quite north, it's one of the coldest parts of the Mediterranean. At the end of winter time, right? We collected at the end of winter time. And in Ophiocomina nigra, we don't see any pattern, not at all. It's supposed to be a colder species with affinity for all the waters. It's much more abundant in North Europe, in the Atlantic side, but it's not suffering. It's like quite comfortable. So it's true that we didn't detect patterns of local adaptation, but it's also true that it has a very high levels of genetic diversity. It may be involved in this kind of responses. In the case of Paracentrotus, we wanted to be a little bit further as well, and we decided to take and make the same experiments in a warmer population. So we went to North Castellón in Vinaròs. We collected samples a few days later, but the seawater was already at 20 degrees because it's a warmer area. So we started the experiments at 20 degrees. We have the control years and the treatments, and what we see in this warmer population is a different pattern than the one that we saw in North Catalonia. So there's a little decrease in respiration here. It's not so strange. And then a very high increase in respiration rates, but no physiological collapse. So this means that there are different processes affecting the stress responses to temperature in echinoderms. It depends on the distribution range and evolutionary history of the species, but it also depends on the thermal population history, as we saw in Paracentrotus, and also depend on the local adaptation patterns we suspect that from the results with Ophiocombina nigra. Right? And it's interesting analyzing patterns of respiration and adaptation in temperature, but what I like much more is analyzing patterns of adaptation to pH in CO2 vents. Why I like much more? Because for temperature, we are working with populations that are quite far away to each other, right? So there are many other factors that can be affecting our results. Whereas here, in the CO2 vents, the distances are very short. So we have been working for 6 years already in Fuencaliente. This is Fuencaliente is in La Palma Island, here in the south of La Palma Island in the Canary Islands, and you have here a map of the CO2 vent area. So there are 3 emission points of CO2 in this vent, here, here, and here. And at these points, the pH is 7.4, which is extremely low because ambient conditions or control conditions are 8.1. 8.0, 8.1. So it's extremely low. But even so, we have different echinoderm species living here, different starfish and different sea urchins, right? Then we have a transitional area with pH between 7.4, 7.6, 7.8, and ambient conditions. And if you see the scale here, it's less than 200 meters, right? It's nothing. For sea urchins or starfish, it's nothing. It's the length of this room. It's nothing. They can even walk from place to place, but they don't do it. Because differences in pH are an actual barrier for them, right? We have a picture here of the area. The vents emissions area are here. The emission point is quite shallow, so we can collect the samples by snorkeling. We have the lighthouse lab, which is our lab for working here. And this is an underwater picture where you can see a transitional area from normal pH to lower pH. Right? You can see the difference at the whole ecosystem. So Sara González Delgado, during her PhD, was analyzing also random SNPs across the whole genome of Arbacea, and she identified over 400 SNPs under selection for pH. And as I explained you before, here we have the structure for neutral markers. No structure. We have a very rare individuals here, only 2. We don't know what happened with them. But anyway, we cannot see any structure along the 2 emission points, 2 transition points, and the controls. However, we can see a very clear pattern of adaptation with different structure along this gradient, right? And some of the genes, candidate genes under selection of pH, we speak about candidate genes under selection because the reviewers always ask for that, but we are sure they're under selection. So there are at least 2 very interesting genes with annotation that are related with biomineralization and growth, right? So they are related with the animals growing. And similar patterns have been found as well in Paracentrotus lividus, because both species are living in this bent area, right? So in this case, we have more markers, less number of candidates under selection, but what is important is not the number of markers, is the patterns that we obtain. Here there is not a structure, no, all populations are equal, and here we find differences in structure. So there are very strong patterns of adaptation along 150 meters. Nothing from here to there. The populations are completely different, right? Well, not completely, but quite different. So we know that there are significant changes in allele frequencies in these sea urchins, but what about gene expression? Do they have different levels of gene expression or no? Because different levels of gene expression at the end means different physiological performance, right? So to answer these questions, Vanessa Rand, in her postdoc Marie Curie, she was analyzing transcriptomic patterns and levels of gene expression, different levels of gene expression from individuals collected in the emission point at the bed and ambient or controlled individuals, right? As you can see here, she identified over 1,000 genes differentially expressed between controlled individuals and bent individuals. And some of the gene upregulated genes are quite interesting because some of them they were related with oxidoreduction, responses to acidic pH, with the fatty acid and lipid metabolisms, because we suspect that the bent area animals need more energy to maintain internal homeostasis, right? And also higher levels of collagen. And why this is important? This is important we realize a little bit later when analyzing the hardness of the skeletons of animals from bent and control conditions, because we saw that animals in the bent are smaller, but the skeleton is harder. But it's quite surprising because they cannot calcify as well as the ambient animals. What is happening is that they are upregulated collagen. They have more collagen in the skeleton. More collagen in the skeleton means a stronger skeletons. That's why in the bend they are smaller but harder. They cannot calcify as well, but they have more collagen in their skeleton. Right? And a similar pattern in the case of Paracentrotus lividus. In Paracentrotus lividus, we have 191 genes differentially expressed, right? Then we analyze as well levels of stress from oxygen consumption, and surprisingly, there are not stress in animals living in the bent condition. Why? They are not under stress, because they are very well adapted at the conditions in the bent. They have the physiology completely adjusted to the bent conditions, to the low pH, right? So you have a look here to oxygen consumption, animals from ambient conditions to ambient points, and the bent condition, the respiration rates are equal, and the same for Arbacea. So ambient, bent, and bent condition. But are these differences in gene expression constitutive, or they are the results of plasticity? What do you think? How can we know that? We decided to make experiments, right? Then we took, sorry, no, I don't want to, yes, it's fine. So here, I'm back for a second. So here you have the, in Paracentrotus, I'm going to show you for Paracentrotus. Here you have the levels of gene expression levels for bend and control conditions. So I'm going to use this, the same picture in here, right? So it's this one. So here we have ambient individuals with this blue star in ambient water in blue. And here we have the bent individuals in pink, the star in pink, with purple and bubbling water, right? And we have that pattern of transcriptomic patterns, right? And what we made later was collecting samples from the ambient condition and put that individuals at low pH with water from the vent, which is this one. Ambient individuals in vent water, right? It's the same here. Ambient individuals in vent water, acidic water. So so initial hypothesis, if changes in gene expression are due to plasticity, we should expect here the same number of differentially expressed genes than here. Yes? It's clear? Okay. Then if changes in gene expression are due to plasticity, because here we have the same environmental conditions, acidic water, the purple bubbling, and if the genotype is not involved in that responses, we should expect zero differences between these two conditions, right? Is that clear? Well, it didn't happen like that. It was the opposite, right? So ambient individuals in acidic water, they don't have much capacity to respond. We only detected 74 genes differentially expressed. It's almost nothing, right? Whereas in this case, we have a high number of genes differentially expressed when we expected 0. So that means that the genotype is even more important than the environmental conditions to determine the expression patterns, right? So, no, these patterns of gene expression that we found between ambient animals and vent animals are not due to plasticity, right? They are adaptive. So these animals living in the vent and control condition and across the transition, they have very clear adaptation based on allele frequencies, significant shifts in allele frequencies, and also different levels of gene expression. And if we have, it's current, if we have a look to the oxygen consumption levels. Here we have the ambient conditions, vent conditions, no experiments, no changing environmental conditions, and here we have ambient individuals in acidic water. Forget about this one. As you can see, the levels of stress increase it. So, and finally, I'm almost at the end, so you can be relaxed. So I have been talking about intraspecific processes, but I wanted you to know that we have been also working on the interaction of the host and the bacterial, internal bacterial. We have been analyzing the symbiotic bacterials in coelomic fluid inside the animals. It's equivalent to our blood. And they have a lot of symbiotic bacteria inside, and they are different between vent and control conditions. Here you have Arbacillixula, here Paracentrotus, and it's different from the surrounding seawater as well, right? We have been also analyzing that for larvae, and we saw large differences between ambient conditions and vent conditions as well. And I would like to highlight how difficult it is working with larvae, because first you need to collect the animals, spawning, taking the gametes, fertilization, and then taking care of the babies and feeding them, right? And all of that without killing adults, because we don't kill any animal from the vent area, the vent systems, right? So what can conclude from the data that we have today? That most echinoderms have pattern or local adaptation, most of them, not all of them, which means that each population is unique, right? So the species respond to rising temperatures are influenced by a combination of different factors, including the distribution range of the species, thermal population history, patterns of local adaptation, etc. Right? And in the case of the vent area, the different patterns of expression cannot interpret as responses to stress, but animals in highly adaptive populations may have reduced plasticity. So what is, for me, the take-home message is the echinoderms highly distributed across natural gradients may already exceed pre-adaptation to future oceanographic conditions, but it's important preserving the current genomic and genotypic diversity for the long-term survival and resilience. And that's all from me. Thank you very much for the whole team. This is Sarah taking some samples in the vent area by snorkeling. And if you have any questions, they are welcome. Thank you.
We have time, plenty of time for questions.
I can speak loud. Okay.
Demasiado corto.
So thank you very much for this talk. Really interesting line of research. I'm also very interested in population differences and how each respond to warming, so I really like it. And I think I have two main questions as a non-physiologist.
I'm not a physiologist as well.
No, but some, measuring oxygen, and I was wondering, how do you infer, or in what level you suggest that an animal is stressed by measuring the oxygen, because we know that cold-blooded species, animals, should increase their oxygen consumption as it gets warmer, right? A pattern that you not every time see. I'm just wondering how.
Well, when you see signs of stress, the animals move less, and they are like, you see that they are not feeling quite well. That's why we interpret that increasing in oxygen consumption is a measure of stress. And there is also plenty of papers explaining that that kind of increasing in oxygen consumption is an indirect measure of stress. Because when we are under stress, we increase metabolism, and we also increase our oxygen consumption.
Yeah, okay.
And also because we acclimatized the animals before. We have them for 2 days under controlled conditions, quite stable conditions.
Yeah, I was wondering, when we see this oxygen increase, is it really stress? Or maybe, you've shown that you have another PhD that maybe measures the blood, right? Or something similar. Is it possible also to measure cortisol? Is it relevant in those animals? Cortisol is a measure of stress.
It has been made in starfish, but we don't measure cortisol. In other species, they also measure cortisol, and it's very correlated oxygen increasing in oxygen consumption with the increasing in cortisol.
And another tiny question, just wondering about this extreme pH gradient. Did you say what causes this gradient? I missed it maybe, or why there is such an extreme pH gradient in such a small scale?
Because the CO2 vents, they are emission points of CO2.
Okay.
On the
Volcano, or because of why is
Are you asking me about the geological reason of
yeah,
that?
yeah.
Yes.
What
Sorry,
causes this
I
extreme
did.
gradient? That's it. Is it CO2 that?
Is the CO2 mixing with the water? Yes.
It's CO2 that comes
It's
below
volcanic.
the sediment?
It's volcanic.
Okay.
So
very cool.
I don't have the picture here, but it's so that CO2 came from the volcano, and they mix inside the rocks, and then they release all the acidic water. Seawater, acidic water. That's why we have this strong gradient.
Thanks.
Thanks for the talk. I want to ask, you show that in one species, when you measure how change the oxygen consumption in function of temperature, in one species don't increase the oxygen.
Yeah.
And I understand that it's
It
weird.
was a little
Not
increase,
weird,
but no significant.
Okay.
Yeah.
Because I imagine that if we increase more temperature, at one point the oxygen consumption should increase at one point in this species, but it's weird because it's a cold adapted, I understand.
Yes.
Do you have information about the thermal limits about this species?
No.
Because
No.
I imagine
Actually, we were looking for them.
Okay.
They exist for sure, but we didn't have any idea. There is not much research about that. And also, we wanted to keep our study quite realistic, because we may increase temperature at 36, but it doesn't make sense, right? It depends on the question. If you are looking, if you are only looking for the thermal limits, yes, you can keep increasing and increasing. But we also wanted to keep it a little bit realistic.
More questions?
Hola, Rocío. I love the presentation. Thank you so much. I was most interested in the bench with the echinoderms. I love the setting and the area. You were mentioning that in your experiments, you were like mimicking like kind of a translocation, right?
It's not actually a translocation because we don't want to translocate any animals inside the area. As well, we don't want to sacrifice any animal. We made in the lab.
But it's kind of, if you were translocating animals...
It will be quite similar because the experiments...
They will survive, right? Kind of. Or they are surviving.
Difficult to say, yeah. Because in these experiments, so I didn't want to extend it that much, but in these experiments, we actually collected animals and we had it in the lab for at least 3 days, in this case, replicating the natural conditions. So in this case as well, we had animals 3 days in the lab, because otherwise you cannot compare if you just measure animals when you took it from the sea. You need to replicate the same conditions for all of them.
Mm-hmm. But yeah, my question would be, so what would happen for the larvae? Have you tried, I mean, if the adults more or less can survive the change?
Yeah, they more or less can
Have you
survive.
tried with the larvae? I mean, they can settle?
We are on the process. We are on the process. We have been cultivating larvae in the lab, but only for 3 weeks. After that, all of them die, because you have to feed them. And the food is different depending on the larvae state. So the maximum for us is 3 weeks. Also because we are working in the lighthouse lab. We don't have much infrastructure there. We need to bring everything from Tenerife in the foray. And it's a little bit difficult because in La Palma there is nice people and all of that, but there is not infrastructure at all. So having dry ice, which is very common here is a nightmare because you need to buy the dry ice in Tenerife, put it in the ferry and bring it to La Palma. But in Tenerife, you can only buy 10 kilos or more. No, you cannot buy a kilo, 2 kilos of dry ice. So it's not so easy. Yes, but the maximum that they survive in the lab, in our lab, is 3 weeks.
Yeah, thanks.
Mm-hmm.
Any other question? I have a question.
Obviously.
Because you said that Paracentrotus, if you increase the temperature to 26, which is what happens in the summer, right? They have a physiological collapse.
Yes.
So that means all Paracentrotus in the Mediterranean in the summer are...
No, exactly, because we are increasing temperature very fast. And this is not happening in summertime. But the increasing is slowly. But they are very close to the limit, the physiological limit. Actually, in the last summer, last year as well, we have seen a lot of sea urchins with gulls, that the infection on the epidermis. Yes.
But there are not massive decimations of the populations, right? Even in the heat waves, can they?
Not really. Not really. They are in the limit. Yep.
Okay. Any other question? No? We can ask her more questions in the kiosco if you want to have a beer with us later. But if not, let's thank again Rocío for her interesting talk and amazing
short
result. And very
because
short.
they asked me to be short, and I have tried to do that.
Yeah. Thank you, Rocío.
Thank you very much.